When studying the bacterial cellular structure, it's very important to know the structure and function of each part. This is crucial if we want to control the growth of bacteria. So, either increase the growth of bacteria for industrial benefits or suppress the growth of bacteria that is pathogenic for us. As you can see in the figure, bacterium cell is surrounded by several layers which are collectively called cell envelope. The cell envelope is defined as the plasma membrane and all the surrounding layers external to it. The cell envelope of many bacteria consists of the plasma membrane, cell wall, and at least one additional layer. In this topic, we will describe the plasma membrane. Keep in mind that bacteria have to interact with their environment in a selective way to acquire nutrient and eliminate waste. This is usually occur through the plasma membrane. Also, a bacteria is no different to a human being. They need nutrient for their growth and they always eliminate their waste. Plasma membranes are an absolute requirement for all living organisms because they carry out the following tasks. First, it encompasses the cytoplasm. Second, it is considered a selectively permeable barrier which allows particular ions and molecules to pass either into the cell or out of the cell, while preventing the movement of others. Third, it interacts with the external environment. To study the plasma membrane, the most widely accepted model for membrane structure is the fluid mosaic model of Singer and Nicholson, which proposes that the membranes are lipid bilayers within which proteins float. As you can see in the figure, you can imagine the lipid bilayer as a carpet where proteins float. The membrane lipids are amphipathic, which means that they are structurally asymmetric, with polar and nonpolar ends. The polar ends are hydrophilic, which means they interact with water. And the nonpolar tails are hydrophobic, which means they are insoluble in water. There are two types of membrane proteins. The first type is the integral proteins. These proteins are embedded inside the plasma membrane. What distinguishes these proteins? First, that they are not easily extracted from the membrane and are insoluble in aqueous solutions. Many are transport proteins used to move materials either into or out of the cell. The next type of protein is the peripheral proteins. These are loosely connected to the membrane and can be easily removed. They are soluble in aqueous solutions and make up about 20 to 30% of total membrane protein. To summarize in this topic, now you know that the fluid mosaic model is the best to describe the plasma membrane. We know now the plasma membrane is made up of lipid bilayer where protein float. Proteins are two groups, either integral or peripheral proteins. Okay, now that you've understood the structure of the plasma membrane, we will next study the different structures of the cell wall. So hang on for another interesting topic of the amazing world of bacteria.